Hello, my name is Ben. I'm from the University of Tennessee, Knoxville in the United States, and I am very excited to tell you about our toolbox for working with high feedback in gene regulatory networks. Well, first, you may wonder, what is high feedback and why is it interesting? High feedback is when multiple feedback loops are interconnected in the regulatory network. This is interesting because it can give rise to dynamics of gene expression that are important for differentiation or development. For example, T cells differentiate into their final state through multiple distinct cell states. Each of these distinct states has a different stable gene expression profile, and in terms of dynamics, we call these different states attractors in the space of gene expression. When there are multiple such attractors, we say the system is multi-stable. Previous work on T-cell differentiation searched for regulatory network topologies that could give rise to this multi-stable, stepwise dynamic. It found two distinct motifs, so-called type 1 and type 2. Type 1 involves three positive feedback loops that have at least one node, one gene, in common, whereas type 2 involves two independent positive feedback loops, the red and blue, that are bridged by a third positive feedback loop. And in terms of graph theory, these loops are called cycles. Because the motifs are defined in terms of cycles, it's all right for there to be some repression as long as the cycle is net positive. T-cell differentiation is not the only process that involves multi-stability. Another important process that does is epithelial mesenchymal transition, which is important, for example, in cancer. The network controlling EMT is quite complex and involves many feedback loops. Feedback loops are also widespread throughout transcriptional control, so it would be ideal if there was a toolkit that could explore high feedback in even larger networks. Now, there are already many algorithms that can search for more specific motifs, but not for motifs that are defined in terms of cycle interconnection. So we sought to create a toolkit that can search for instances of high feedback topologies, quantify enrichment, that is, how much more abundant they are in one network than would be expected by chance, model the dynamics they might give rise to, including sustained oscillation, and visualize all of that information. Starting with detection, our toolbox can search for instances of several different high feedback motifs, including type 1 and type 2, also variants such as mixed sign high feedback that may include negative feedback loops, and more specific type 2 topologies that involve mutual inhibition between the two independent cycles. We call these illustrations logos. They illuminate the cycle interconnection pattern in identified subnetworks. For example, if we are starting with this network, which is a part of the EMT network, and we want to find high feedback topologies, HiLoop might extract this subnetwork. Each cycle is colored differently. We see it has a logo summarizing the two independent feedback loops bridged by this gold cycle. This is indeed a type 2 topology. Extra edges that are not necessary for this topology to meet the motif definition can be retained, but they are colorless. You may notice that the edge from Mir 101 to SNE1 has been doubled. This is done when one regulation is involved in multiple cycles. This makes it easier to trace each cycle. To quantify the enrichment of a network in a motif, it needs to be compared to permutations of the network. Permutations are generated by edge swaps and sign shuffling. Then the proportion of permuted networks that have at least some number of motif instances can be counted, and the proportion of permutations with at least as many motif instances as the input network serves as an empirical p-value for the enrichment. If we find that the EMT network is somewhat enriched in positive feedback loops and very strongly enriched in type 2 topologies. Once you've chosen a topology whose dynamics you would like to explore, HiLoop can convert that subnetwork to a system of ordinary differential equations. The network structure determines how many equations there are and how many of these Hill terms there are, whether they're activation or repression. Then, to explore the range of dynamics, many different parameter sets are sampled. Then each parameterization, each system, is simulated starting from several different sets of starting concentrations. The endpoints of those different simulations are examined for multiple distinct attractors and sustained oscillations. This produces a collection of parameter sets and attractors. To visualize these results, HiLoop generates plots. Here I'll look at possible dynamics of this small mixed-sign high feedback network. The scatter line plot shows the position of every attractor in each system in the space of two genes expressions, or PCA, whereas the cluster heat map has one row for every attractor, and they're all clustered regardless of which system they came from. The colors indicate how high or low each gene's expression is. Looking at all the parts in more detail, on the scatter line plot, attractors from the same system are connected by lines, and in the cluster heat map, they are joined by optional arcs. Arcs of different systems are organized by how many attractors there are in the system and by how many species are monotonically correlated to each other, which is useful when studying differentiation. Looking at where the oscillatory attractors, shown as gradients in the cluster heat map, are, we see that only a few of them are in the same system as another attractor. Many are the only attractor in their system. To more easily see the range of possible unique attractors, similar attractors can be folded together. How many instances there are of each attractor is shown in this strip on the left. Regions of expression space that have very many attractors are also shown with the contour shading. In summary, our toolbox HiLoop can both visualize high feedback subnetworks and the range of dynamics they could produce, serving as a hypothesis generator, and it can perform an enrichment analysis to serve as a quantification method. Using it, we determined that the EMT network is very strongly enriched in a certain high feedback motif. This toolkit is publicly available on GitHub. I would like to acknowledge the authors of previous work and the NIH for funding. I would also like to thank my advisor, Professor Hong, and thank you for listening.